Good morning, everyone. Let's come to one of the important topic of your molecular biology, which is post-transcriptional modification or post-transcriptional gene silencing, which is important topic of your study. Okay, so post-transcriptional gene silencing, what you call TGS, or post-transcriptional gene, or what you call post-transcriptional gene silencing, what's called PTGS. You see, there's a difference between what's called transcriptional gene silencing or post-transcriptional gene silencing. Okay, so here the proteins are silenced here, and when you see in PTGS, promoters are active, genes are hypermethylated, and genes are hypermethylated in the promoter region. Here you have hypermethylated in the coding region. So what is the purpose? You generally will find in the viral immunity, and we still don't know what's the important function, and that's the reason you call it RNA interference. So other times, other names of post-transcriptional gene silencing is PTGS, gene silencing, or RNA silencing, or RNA interference, and certain fungi you call quelling. Okay, RNAi can spread throughout the cell, certain organisms like C. elegans and plants. So what's post-transcriptional gene silencing is the ability of exogenous double-stranded RNA or small RNA to suppress the expression of a gene which corresponding to the double-stranded sequence. Okay, double-stranded RNAs are generally produced by, of course, transcription of inverted repeats or viral replication. Okay, or transcription of RNA by RNA-dependent RNA polymerases, or double-stranded RNA triggers cleavage of homologous mRNA, or PTGS is defective. Plants are more sensitive to infection by RNA viruses. So in RNA interference defective nematodes, for example, transposons are much more active. So you see RNA can be induced by C. elegans. Okay, you take a double-stranded DNA. Okay, you soak it with the C. elegans and you inject double-stranded. Now what you do is you can use that RNA or double-stranded RNA in such a way so you can inject into the C. elegans to stop the expression of certain kind of RNA, and so you could do for the Drosophila too. And of course you can use it in mammalian cultured cells also. So what's the mechanism of RNA interference? So that's why you call RNA interference. You see, there's double-stranded DNA which has to be broken down into small pieces, what's called the enzymes, what you call Dicers enzyme, where you take a dsRNA to cut down into small fragments, which is 21 to 25 nucleotides long. Then you use there is one complex called RISC complex for what's called RNA-induced silencing complexes. That's RNA-induced silencing complexes. Okay, along with this RNA, when it is targeting when it's targeting one of the protein or one of the RNA, homologous mRNA target recognition, it helps in silencing of the RNA. So this is a whole mechanism. When you see a double-stranded RNA is there, it's coming inside with the help of the dicer. Okay, it's making a complex along with the RISC complex, and it's targeting small fragments of the RNA. Then you see miRNA, microRNA complex has been formed, and throughout this process the RNA is silenced. So you see that cellular response to presence of double-stranded RNA that results in the sequence-specific degradation. So you can use a specific, for example, any cell which is coding for the RNA which is involved in the cancer. You can recognize such kind of elements in the DNA element in such a way it will recognize that element and it will sit on that element so as to stop the expression of the RNA. So you see the mechanism of RNA interference. You have a dicer enzyme, as I was saying, that cuts the dsRNA into 20 different fragments. Now it produces a small siRNAs. This siRNA, along with the RISC complex, what's called RNA-induced silencing complex, which is a multi-protein machine that binds to the RNA. Okay, and with the RISC, with this, along with the single-stranded RNA, it will sit. It will be directed towards mRNA where it has to be targeted, and this will induce the silencing of that particular mRNA. And after that, the RNA catalyzes the cleavage of the mRNA and so on. And this is the way you can silence certain kind of RNAs. Okay, and that's what you have. So many RNAs has been destroyed in such a way that it's making so many. The says that the RNA is no more functional. Okay, I hope you understood a basic concept of RNA silencing, what you are finding. Okay, so what are the important applications of RNA RNAi or RNA interference? For example, modulation of HIV is important, replication of RNA interference, or potent or specific inhibition of human immunodeficiency viruses. Okay, and you can induce RNA interference in adult mice to study different types of diseases like diabetes or neurodegenerative diseases. Okay, and or metabolic diseases, and so cancers or any kind of new metabolic diseases, or successful inactivation of oocytes and so on, or developmental diseases. So possible future improvements of RNA implication, RNAi application is if you can come up with U6 RNA promoter based plasmids, and it's potentially or in vitro synthesis of T7 RNA polymerases, but potentially it is useful in creation of siRNA vectors with resistant cassettes and establishment of inducible siRNA or establishment of retroviral siRNA vectors. Okay, so I hope you understood the basic concept of SI or RNA silencing today, with a special focus on single interfering RNAs, siRNAs, and so on. Thank you so much for your attention.